Hello, my name is Yvonne, and my project is called Upgrading the Dendritic Cell, CARs on Dendritic Cells for Improved Cross-Presentation and Anti-Cancer Immunity. And so, as you might be able to guess from the title, dendritic cells they're important for anti-cancer immunity, and they're an immune cell. And so, if we really quickly think about how cancer is normally cleared in the body, we think of our cancer-killing T cells that go and attack our cancer cells, which leads to that cancer dying. But if we ask ourselves the question, how do those T cells actually know to target cancer? That's where dendritic cells play a really important role. And so, say we have a tumor on our arm, for example. What would happen is a dendritic cell would come along, start nibbling at that tumor, and take that ingested tumor material and show it to a T cell. And so that is the actual process that converts a resting, naive T cell into a cancer-killing T cell. It's a dendritic cell actually showing a T cell what that cancer looks like, what it tastes like, kind of thing, which prompts it to want to kill that cancer cell. And so my project looked at how can we make the dendritic cell even better at its job. And so chimeric antigen receptors, or CARs for short, are one way that we can genetically modify a cell to enhance its natural function. And so with our CARs, we tried to design it to increase antigen uptake, or basically the amount of tumor material that it can ingest, and cross-presentation. And cross-presentation is just a term that we use to describe how many T cells can we activate. And so our hypothesis was that the dendritic cells with our CAR would have increased antigen uptake and cross-presentation compared to a wild-type dendritic cell. So now how do we actually measure if our CAR can actually help with antigen uptake? And so to do this, we had dendritic cells with our CAR and without the CAR, and we'd feed them both tumors. We would let the dendritic cell and the tumor incubate for 2 hours, and then after 2 hours we would look under a microscope and quantify how many of each cell type was eating the tumor. And what we find is that the dendritic cells with our CAR, they eat a lot more of those tumors than our wild type does. So in conclusion, we can say dendritic cells with our CAR can significantly increase the amount of tumor material that they ingest. But now we want to know, really importantly, is that do these dendritic cells with CARs, do they activate more T cells? And so to compare the amount of T cell activation between the wild-type dendritic cells and the dendritic cells with the CAR, we added tumors to both sets of dendritic cells. We incubated it for 2 hours, and then we added T cells. And so this would allow all the ingested tumor material in the dendritic cells to be shown to the T cells. And then we measured subsequent T cell activation. And so activated T cells would produce a cytokine known as interferon gamma. And so that's what this assay is actually looking for. It's the, it quantifies the amount of interferon gamma using an ELISA. And so what we actually see is that the dendritic cells with our CAR, they actually activate a lot more of those T cells because a lot more of that interferon gamma is being produced with the dendritic cells with the CAR, the ones in blue here. And that basically proves our hypothesis that the expression of these CARs on dendritic cells will increase antigen uptake and cross-presentation. And with that information, we can also speculate that this could potentially lead to a more robust and effective anti-cancer response. And so my future directions would be investigating how that CAR actually improves cross-presentation, the mechanisms, as well as the application of this CAR to existing cancer therapies. So I'd like to thank my lab and my great mentors for a really great summer.